In this module, we will see the structure of a DNA, and we will see how the DNA is coiled and it's recoiled to form the structure of into the chromosome. So to see this in the little bit detail, so I will discuss the DNA structure. Here we can see the DNA coiling effect and of a molecule of DNA that bound together with the proteins called histones, and these proteins are further coiled to form the small circular structures called nucleosomes. So these nucleosomes further coiled and form the condensed structure called the chromosome. So chromosomes store the genetic information. In humans there are 46 chromosomes, and we get 23 chromosomes from the mother and 23 chromosomes from the father. So chromosome contain all the genetic information that is passing on to the offsprings. For example, eye color, skin color, height, and all the characters we attain. So all information is stored in these chromosomes that are coiled and condensed molecules of DNA. DNA has specific structure called double helix. So double helix, we can see here, it is double helix. Double helix describes the structure of a ribbon that has internal extensions. So we can see here internal extensions and are attached to a mirror image of ribbon on the other, bonded to the extension coming out internally, and create a twisted, you can see here twisted structures. These are called double helix. So double helix of DNA consists of molecules called ribose plus phosphate molecules, and these form the sugar phosphate backbone. And then these sections called nucleotide bases. So these sugar plus phosphate and the nucleotides, they consist to form the nucleotide. So nucleotides consist of 4 bases. One is adenine. This is adenine, and we can also represent them with A. Other is thymine, represent them with T. The third one is guanine with G and cytosine with C. So we can see here adenine will always bind with thymine and guanine will always bind with cytosine. So adenine, for example, and thymine, they form to join together with the phosphate and sugar bond to form the nucleotide. These nucleotide bases only bond with specific nucleotide bases, as I told earlier. So DNA molecule has a backbone of sugar and phosphate. So here we can see in detail the molecular structure of the DNA. Here we can see again the backbone structure consisting of phosphate and the sugar molecule. And on the other side of the DNA molecule, again we can see the sugar and the phosphate molecules. The backbone is attached to the, going towards the other side of the double helix is the nucleotide base. So we have discussed that these all composed to phosphate and sugar molecule, and phosphate and sugar on the other side, and 2 nucleotide bases, they consist to form the nucleotide. The backbone is attached to that, going towards the other side of the double helix is the nucleotide base cytosine or guanine. So we can see here the another feature of this DNA structure is the hydrogen bonding. So we can see the guanine and cytosine, they are attached, linked together through a hydrogen bonds. If there is a guanine, it will bind with the cytosine. One section of the double helix would be sugar and the phosphate, as I have described earlier. This consists 1 unit of DNA double helix. So there are only 4 possibilities in the DNA structure. Either it will be thymine that bind with adenine, or it will be cytosine that will bind with guanine, or it will be adenine that will bind with thymine. So overall we can see here that one nucleotide, this is one nucleotide, we can see here that this is the phosphate sugar molecule, then there will be a one nucleotide attached with the other nucleotide, and then again the sugar and phosphate molecule. So again we can see the phosphate and sugar molecule attached with one nucleotide, and it is complementary bind with the other nucleotide, and it will be sugar and phosphate molecule to comprise the nucleotide bases. So only there are four possibilities. Either it will be guanine cytosine, thymine adenine, cytosine guanine, and adenine to thiamine.